Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute.
From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on December 4th, 2024. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 70th episode of Matters Microbial. Wow. Yet another milestone for this podcast idea of mine. I'm doing my best to present microbiologists and their work to a diverse audience. And I appreciate every listener, every viewer, as well as every email and comment. And of course, my guests. Here at the University of Puget Sound, we're rapidly approaching the end of the semester. The Micronauts in my microbiology class are working very hard. Relevant in some ways to today's topic, two of my Micronauts identified a bacterium that has antimicrobial effect on E. coli, as you can see from the zone of inhibition here. That same isolate also inhibits Bacillus subtilis, as you can see here. And finally, it also inhibits Acinetobacter, as seen here. That's quite a broad spectrum. What my students found most interesting is that their isolate was fluorescent under ultraviolet light, seen here. This may be because of siderophores that are binding iron in the media. We hope to learn the identity of this isolate via 16S ribosomal RNA analysis by next week. I will keep you posted. Remember, students found this isolate from a moss sample here on campus. We do not know the nature of the antimicrobial effect, but we will be sending the strain to the Tiny Earth Consortium for further analysis. My students are also studying a metagenomic phosmid library made from a soil sample. We're looking for antibiotic resistance genes. Now, we'll talk about that in coming weeks, but here, on this XGAL plate, students found a blue colony. That particular phosmid has an insert from that particular soil sample that possesses a gene with beta-galactosidase activity, and we don't know from who or where. Thus, I hope to get DNA sequence from these phosmids and begin learning about the source organism of the cloned genes. Again, and I can't emphasize this enough, students are fascinated by things that they find, not exercises that we give them. I also don't avoid the artistic when it comes to microbiology. Here is a Lux portrait of Edgar Allan Poe, painted with luminous bacteria by my very skilled wife, Dr. Jennifer Quinn. But do not be fooled. Even new practitioners of Lux art can do fabulous work, as seen from the student art here from last year. And this year, we're playing a bit with fluorescent bacteria, that is to say, E. coli expressing fluorescent proteins. Dr. Quinn painted her microbial version of the Scream, something every student can appreciate as final exams loom. This is the next to the last time I'll be teaching microbiology because of my phased retirement from the University of Puget Sound. I am so very grateful to the students who bring creativity, laughter, and wonder to my classroom and lab. I will miss them very much. One topic that I insist my students learn about is the rise of antibiotic resistance and the rise of antibiotic resistance bacteria and the threat that they have become to us. But what is that threat, and what about our practices increase the chances of potentially disease-causing bacteria becoming resistant to antibiotics. My late father was raised on a farm, and he always chided me, city boy, for not knowing much about how food arrived at our table. So I am beyond delighted to welcome Dr. Erika Ganda to the podcast today to discuss the growing threat of antimicrobial resistance, the One Health approach to agriculture, and the fascinating and diverse research conducted by her team of colleagues at Penn State. Welcome to the Quality Quorum today, Erika. It's nice to meet you.
Mark, thank you so much. I'm very excited to be here, and it's a great pleasure to have been invited.
Oh, it's so much fun. And as I say, one of the most important things we can teach students is about antibiotic resistance because it's something that's in everyone's life for good or for ill. And that's why I'm delighted that you agreed to be on the program. But the first thing that I want to say to listeners and viewers is that you're a double doctorate.
That's right, I guess. Yeah.
You
I'm a
are
veterinarian, yeah.
You're a doctor of veterinary medicine as well as a more research-oriented PhD, and that's a lovely combo. So you have a lot of degrees to your name.
No, I don't like to think about it that way. I feel like I just stumbled on things as my career progressed and I ended up here. So I am not a farm kid either. None of my immediate family members had any kind of agricultural background, but I was born and I decided that I loved cows. So that's why I went to vet school because I loved cows. And towards the end of vet school, I got exposed to research for the first time, and that's when I heard about microbiomes. That's where I moved toward bacteria and ended up with my PhD. So things show up in our life, and we never know where we're going to end up.
I have a couple of former undergraduate research students that earned their PhD on rumen microbiology, and so that whole business of they all love the cows. Let's put it that way. And I think that's one. In fact, I see them behind you too on your wall. And so that's a wonderful thing. So I think a good thing to discuss now with listeners and viewers is what the term One Health means.
That's a fun thing because being a veterinarian, One Health is a very commonplace term. And when you start talking to folks not within your veterinary world, you realize that not everyone really gets it until you explain what it is. And they're like, oh, that makes total sense. So, what is One Health? So, when you think about health, you have the health of human beings. We have been very human centric, even though bacteria are the best. Mm, But for you to have healthy human beings, you need to have a healthy environment for these human beings to live. If these human beings are omnivores, you need to have healthy animals for them to eat and have good nutrition. If you have your vegetarians and vegans, you still need a good environment and agriculture to be able to have your soy and your corn and all your veggies. But to me, the glue that actually keeps everything together are the bacteria. So if you think about every fall when all those leaves fall to the ground, if there weren't for all the ecological services that the microbes are giving us, we would be swimming in trash right now.
Mm-hmm.
So really, bacteria are the glue to keep everything together. They help us with nutrient cycling and cleaning up messes that humans make. Another really cool thing in terms of loving cows is the cow superpower is they can take food that is very low in nutritional value if a human were to eat it, which is grass, and they can transform that in very high nutritional value, such as milk and meat. So that is only possible because of the bacteria that live inside the cow's stomach. The cow herself cannot digest any of the food, most of the food that she eats. Another fun fact with bovines is you don't really have to balance the diet for some cows for certain kinds of vitamins because the bacteria living inside them actually make all the vitamins that they need. And then to close the loop really is this flow of microbes that help us produce our food, keep our environment clean, have our nutrient cycling that really keeps this one health concept where if you want to have healthy human beings, you also need healthy environment and healthy animals and a healthy agricultural system. So that's the concept of one health right there.
I think it's so well put because we tend to be separated, and that's what my father and his father before him used to insist to me, that food didn't appear in a grocery store, and how that took place was very important. And it makes perfect sense to hear you say that if we want to be healthy, we have to make sure that the food that we produce was produced in a healthful way as well. I love your idea about bacteria being the glue of everything, because they are. I certainly am microbially centric. That's what it comes down to myself. But what I would say is that they are the basis of the very biosphere. And it's really interesting because many times I can't get folks to really appreciate that. And that's one reason I do this podcast, because I know when I speak to other microbial scientists, I'm with my quality quorum. So that's a really good thing. The other thing I would tell listeners and viewers to remember, because I just got through teaching about this, is the issue of the rumen, which is how the cows are able to actually break down cellulose in this wonderful fashion. It's like a zoo of different types of fungi, protists, bacteria, archaeans, all working together to make this happen. And it's so fascinating to remember that in many cases, the nutrition the cow gets are volatile fatty acids. And that's so different than the way that we take in food. And it's like a walking fermenter, for lack of a better term. That's what the cow's rumen is.
That's exactly right.
You probably have done the routine where there's a particular type of a port that's been installed in cows where you can take a sample, and I have done that.
Mm-hmm.
And I'm as a veterinarian, if nothing else, you've done very similar things, and it is such a zoo inside of a cow. It's so fascinating.
Yeah, the rumen microbiome, it's really exciting, and there are a lot of potential new pathways that are within the rumen that we really don't know much about. So I'm really excited to see what the next research coming out in this field it's going to show us.
So this leads us a little bit to the idea of antimicrobial resistance, and I'm going to put some links in the show notes to some alarming videos about the rise of AMR and the threat that might become with us. And many of us are familiar with the idea that bacteria become resistant to antibiotics by one of several mechanisms. And that just happens because of a numbers game and the mutations that exist in nature. Those that are resistant to an antibiotic can grow, and so they have an advantage. Add to that horizontal gene transfer, and it is a horrific problem. We're used to it in medicine, say by the overprescription of antibiotics or misuse. But in fact, it's relevant to agriculture as well.
That's true, and that's one of the big missions of my lab is to be able to work on that aspect. Um, Everybody needs to eat every day, and we have farmers that work really, really hard to put food on our tables. And I feel like on the issue of antibiotic resistance itself, there is a lot of finger pointing toward
Mm-hmm.
agriculture. Yes, we use a lot, but we also grow a lot of animals.
Mm-hmm.
Farmers don't like to just give antibiotics just because. So, one of the goals of my lab is to be able to educate the general public that yes, we use antibiotics in agriculture. We have many rules and regulations regarding of what antibiotics we can use, and we have something called the withhold period, which is if I have to treat a cow, I have to throw her milk out for a specified amount of time until that drug clears out of her system, and then I can put the milk back to go into the food supply. So that's one of the facts. The other big point that I'd like to raise is that while there is a lot of antibiotic use in agriculture, most of antibiotic resistant infections that end up taking human lives are not necessarily linked to the agricultural system.
Mm-hmm.
They are usually hospital-acquired infections, or you have things like TB. There is a very big chunk of it that is associated with food, which is foodborne pathogens. And one of the pathogens that we like to work on in my lab is Salmonella. So Salmonella on itself, I have a love-hate relationship with, because it is such a mess. It's like two species, but over 2,500 serovirus, and they carry all kinds of weird genes, and you think they're gonna do something in the lab, and then they don't do it, and you see that they're killing animals in the farm, and you go try to isolate it. Sometimes you can't get it. So I'm gonna share a little bit of our Salmonella story. Most folks of my age and older are going to be very familiar with the term typhoid Mary. But when I teach undergraduate students right now, I ask them, do you know who Typhoid Mary was? And that's just blank stares.
Mm-hmm.
So for those who don't know, Typhoid Mary was a cook in the early 1900s that was an asymptomatic carrier of Salmonella typhi, which means she was completely healthy, but she was colonized by Salmonella typhi. And she liked to cook for people. But she wasn't very good at being hygienic or washing her hands, I guess.
Yeah.
So what happened is there was an epidemiological investigation, and they started to trace back people dying of typhoid fever associated with families for which that Mary lady had cooked. So they finally ended up tracing it back to her. And because it was the early 1900s and we did not know too much about microbiology at that point, there was no other solution other than put her on house arrest, because she was told to stop cooking for people several times and she just would go and find another job. She had to
Mm-hmm.
leave. So they put her on house arrest to be able to stop the typhi uh, case outbreaks that were occurring because of her cooking. So why do I say that? Because you have a completely healthy human being walking around other human beings, spreading a bacteria that can be deadly. And in that case, it's Salmonella typhi. Salmonella Dublin is one of the bacteria that we are really interested in my lab. And it is the Salmonella typhi of dairy cows, meaning that
Mm-hmm.
we can have completely healthy animals walking around the farm carrying these bacteria. And whenever you have a challenge, be it a cow is having a baby, or you take the baby from her mom and put it with its mates in a group pen, their immune system gets challenged, and that's where this opportunistic Salmonella Dublin comes up. And one of the things that we work on is, for most folks that have heard of foodborne disease and Salmonella, they will think diarrhea or gastroenteritis. Salmonella Dublin is really interesting because that's not what it causes. There are cases of a gastroenteritis, but the big symptom of Salmonella Dublin infection is respiratory disease in dairy cows, dairy calves. And the strain that is circulating in North America is carrying a plasmid that has lots of antibiotic resistance genes. And to make matters even more complicated, as I mentioned before, we have lots of rules and regulations we have to follow in terms of treating food animals with antibiotics. The strain of Salmonella Dublin circulating in the North America is naturally resistant to most antibiotics that are approved for treatment of respiratory disease in baby cows. Salmonella is a poster child of a One Health challenge because it's a animal adapted pathogen that can survive for a long time in the environment and can cause disease in humans. And when it causes disease in humans, is extremely severe. So it's a very big problem on that sense. Another fact is this is a zoonotic pathogen. So there have been outbreaks of Salmonella Dublin, making people ill, associated with beef consumption, raw meat. It is a very big occupational hazard because we have the farm workers working directly with the sick animals that are shedding the bacteria. And since it's a naturally multi-drug resistant bacteria, we have very few drug choices to treat these people if they end up sick with this bug. Salmonella Dublin is also very invasive. So the hospitalization ratios for it are a bit higher, and it's just a very fascinating bacteria to study. So
Ca-
we know all of this. Go ahead.
What I was going to say is let me bring a couple of things together for listeners and viewers, because you've said so many things that I think are really important for people to remember. So the first, the idea of Typhoid Mary is super, because that is something that we all need to talk about, because there are some individuals that are asymptomatic but are still able to transmit that pathogen. And that is a really interesting idea. From Mary's point of view, these people were just getting sick. It had nothing to do with her because she was feeling fine. So they told her not to work for these people, but she wanted to work and do something. So she would just go work someplace else. And that's why, and I'm going to put a link in the show notes to that whole story because it's a fascinating one. The other thing that I was going to say is I love the idea of this. I want to be very fair in that one of the things that I worry about are the way that we look at all microbes as being negative. I call those devil microbes, as we often say that things are always positive. Those are angel microbes. Neither of those things are true. It's always context. And it is very easy for people who are not advanced micronauts to point fingers of blame, as you point out. And I hope I didn't give that impression about agriculture. There are many changes, and on your website, actually, you talk about some of the regulatory changes that people make in response to some of the challenges that you face. And I really appreciate the way that you run the balance between the consumer and the agriculturalist in producing things, because this is going to be true no matter what type of food item you're trying to produce. You're still going to have these issues that we have to be cautious about. And what you've done in talking about this has reassured me a little bit. The final thing I want to say is I have to laugh at your comment about how complicated Salmonella is genetically. And the reason so is when I show students that pangenome model, where genes are being acquired and lost, and it's no longer a family tree. It looks like kudzu growing everywhere. And I think it's delightful, but no student enjoys seeing that. And I can't imagine working on it, so applause for you.
Thank you. Yeah, Salmonella Dublin is a bit different on the fact that it is very clonal. So there's very little variability on the strains that we have sequenced, at least in the United States. And that's very different from other kinds of Salmonella serotypes. So yes, Salmonella is a mess. Salmonella Dublin is a little bit of a tighter mess from a genetic perspective. Salmonella Dublin is a real mess when it comes to surveillance, detection, and mitigation. So things that we're working on are, we have farmers that have challenges with Salmonella Dublin. It's killing their calves. And we go out to the farms and we know the Salmonella Dublin is in the environment. We swab, we do enrichments, we try to grow it in the lab, and we can't get it. So it is an opportunistic pathogen from the sense that they can be completely healthy and they're not going to be shedding the Salmonella at all times. So that's a huge challenge for us to be able to find where this shedding event is occurring. The other thing that makes Salmonella Dublin a bit of a worse pain is it doesn't really like our standard procedures for enrichment of Salmonella, because we can find many different kinds of Salmonella in the environment, right? It's the environmental microbe. And when you get a sample that has Salmonella Dublin and other Salmonella, Salmonella Dublin loses the fight in the lab and we never end up finding it. So that leads to a big challenge on diagnostics of Salmonella Dublin as well. We're trying to work on that area. And then the third point is these diseases have been affecting US dairy cattle for a long time. It used to be a bigger problem on Western dairies, larger dairies, but it is slowly creeping its way east. And there is not a lot of research on how widespread this is on the northeastern United States. So that's one of the projects we're working on. Try to work directly with farmers facing the disease, but actually bringing awareness that, hey, you should have this on your radar. It is not a typical salmonellosis case presentation, so you should be asking for those kinds of tests if you have this kind of problems at your farm and so forth. The other thing regarding antibiotic resistance that we work on, and I thank you for highlighting that, is that balance between what we do in agriculture to do best by our animals and keep within the regulations and also respond to consumer pressure. Because in the end of the story, agriculture is a business and we need to have consumers. Lately, there have been a lot of push from consumers saying that they want to only buy products that were raised without antibiotics ever.
Hmm.
Whenever you go to a grocery store, you see all this advertisement of no antibiotics ever, raised without antibiotics. And as a veterinarian, that is a big red flag to me
Mhm.
because I want to be able to treat the animals when they need treatment. So I think this big consumer pressure for antibiotic-free products might be associated with lots of animal welfare issues. Because if I am working on an antibiotic-free system and my animals start getting sick, I'm going to try to wait until the very last minute to treat these animals because then I cannot sell that product at a premium anymore. So the
Erika,
consumers need, go ahead.
all I was going to say to you is that this is a really common idea. How many times do we see things in the supermarket or the fancy market? I call the fancy markets the museum of food. That's the really expensive places to go, like
Mm-hmm.
Whole Paycheck instead of Whole Food. That's mean of me to say, but you know what I mean. And they'll be advertised as being antibiotic free, or non-hormone treated or whatever it is. I've even seen water labeled as gluten free. Now, the reason all of this takes place is not factual. It's about marketing. Now, I'm all for labeling, but that labeling has to be accurate in my view. So, for example, when people talk about organic vegetables, and I've actually seen things labeled as chemical free, and I'm thinking, well, what are these items made of? Happy thoughts? What they mean, is they weren't using pesticides, is what they're saying. But everyone has seen the reports that suggest that not always is that labeling accurate. And so I really appreciate your common sense attitudes about animal husbandry, and I would apply that also to crops. That I think labeling is important, but that labeling needs to be accurate, and it's up to us as science communicators in a way, because that's what all research really needs to make sure that the public understands what we're talking about. And gluten-free water is a silly thing, right? Because obviously there's no gluten in water, but it's there because the people who made the bottled water want you to buy it. So this is really good to get both sides of the issue here. And it's not a negative thing at all. I don't mind people making choices that, for example, I wouldn't make as long as everything is clearly labeled.
Exactly. And I have a recent learning on this gluten-free water because I shared your sentiments with like, what the heck, this is corn, corn is gluten-free. Except I have family members that were recently diagnosed with celiac disease,
Yes.
and those labeling come from a cross-contamination perspective. So that is actually a very important thing that I only learned after being exposed to somebody who will will get very sick if they end up eating oats that had been processed or harvested from a field that had wheat before. So it is a fine balance on what should go in the label and what should not go in the label, but it all boils down to people being able to critically think, why is this here?
Yes.
And does this make any sense? Does it make any sense that I have a chemical-free or a gluten-free salt? Maybe it does, maybe it doesn't. So back to the whole antibiotic-free, especially I'm going to pick on poultry because it's such a huge part of our diet. And it's a very fast production cycle. They are very efficient at producing high quality protein. It's lean. And so I'm going to pick on poultry. And what happened is there was a lot of consumer pressure recently on wanting no antibiotics ever chickens. And one of the things that we are studying in my lab is what are the replacements that are not antibiotics, but something else that people are selling with the claim that it will help animals gain more weight, even though it's not an antibiotic. So a bit of history and regulation. In the past, it was allowable to feed low doses of antibiotics in animal feed for growth promotion. And that it was useful for a variety of reasons, like you decrease infection pressure and such, but it does contribute to antibiotic resistance. And there are a few instances of very clear associations of antibiotics being used in poultry, and then infections that are resistant to those antibiotics showing up, and then you pass regulation to stop the use of that in poultry, and then the resistant infections go down. So since 2017, the Food and Drug Administration passed the Veterinary Feed Directive, and they said, okay, we know better than this. We should not be feeding antibiotics for growth promotion purposes only. So this is completely illegal in this country. However, it's still done widely in many other countries. And as we know, microbes have no borders. If we learned anything from our previous pandemic, is whatever is happening across the world today can be affecting us in 2 weeks.
Yes.
So even though the US is at the forefront in terms of regulation, we are not free from those risks because we travel, we import food, and so forth. But what that did to the US producers is they had a tool that they used to keep their animals healthier and make more profit. Now they have a big gap. And what are they going to fill that with? So there are lots of feed supplements that started popping up in the market, different prebiotics, different probiotics, essential oils. And the thought is, oh, I'm going to feed this to my animals because it's natural. But so it's arsenic. Until we get data that proves that feeding these compounds is more beneficial from an antibiotic resistance perspective, you can't really say that that's any better than antibiotics. In fact, there are studies showing that organic production systems don't have much difference in terms of what antimicrobial resistance genes are circulating among them, because antimicrobial resistance is a natural phenomenon. So now I'm going to call the video from Harvard. Do you know which video I'm talking about? The 1 minute mega petri dish?
That's right. That's Microbiomes is doing the narration. So, folks, what this is work that's been done. It was originally done by Roy Coscioni and Michael Boehm. Michael Boehm is a professor at Harvard now, and it's wonderful. It's a gigantic, ginormous petri dish. And what's taking place is that there are gradations in the amount of antibiotics. And so what you can actually see physically over 11 days is how bacteria are able to mutate to become resistant to sequentially higher levels of antibiotics. None of this has to do with horizontal gene transfer. It's taking place all the time in the space of 11 days so that they'll become resistant to 1,000 times the level of a specific antibiotic. I will certainly put a link to that video. If it's playing in the background, that's wondrous. And also a link to that article, because I promise you this is something that's a must read. I have a saying, and I want you to forgive me for this, Erika, because I talk too much, but I say the hand of Darwin is on all that lives. And it really is. And it's irrational to think that you wouldn't consistently get random mutations. And if there's any advantage to that random mutation, it's going to predominate. And that particular megaplate that you're describing is a wonderful, viscerally impressive example of it.
And I really love to show that video to my students. So I hope our viewers can appreciate how cool that is for several reasons. One, it's evolution happening in front of our eyes.
Yes.
Two, a lot of my students have many challenges in interpreting phylogenetic trees. And then when they see that video and they see the lines connecting the dots, I'm like, this is all that a phylogenetic tree is. It's like a family tree of bacteria, and you just watched it happen in front of your eyes. But that's only one mechanism that antimicrobial resistance arises, right? We have the random mutations, but the other problem that we have is horizontal gene transfer. So sometimes you can pressure bacteria to with a higher salt content, or you put more zinc on their diet that has been known to be antimicrobial properties. Sometimes this pressure can also lead to increased antibiotic resistance because certain mechanisms such as efflux pumps will pump anything that's not good for the cell out of the cell. So I am feeding zinc to make sure my animals are healthier, and this is not an antibiotic, but guess what? I'm still increasing antibiotic resistance genes because the pumps that are being used to pump out the zinc can also pump out antibiotics. Because of mobile elements, sometimes you put pressure on the bacteria with a non-antibiotic. I'm picking on zinc just for sake of examples. But that gene is on the same mobile genetic element as a beta-lactamase, for example. That has nothing to do with zinc. But if you're putting lots of zinc on your bacteria and they're growing and they're selecting for that gene, you might be unknowingly selecting for other antimicrobial resistance genes. And we saw this in one of our papers. It was not causal, it was all correlational, but there was a mine dam that ruptured from like iron mining in Brazil, and that contaminated lots of waterways in the north, what the southeast Brazil. And you can see on the slide here where that area happened was a big ecological disaster that happened in 2015, and that dumped millions of gallons of heavy metal contaminated water into waterways. So what some collaborators in Brazil did is a very simple surveillance and collected samples from two farms, one from cows that were drinking from the contaminated water and another one from cows that were not drinking from that contaminated water. And what we saw was that phenomenon of samples from animals drinking from heavy metal contaminated water had higher levels of antimicrobial resistance. And we know through mechanistic experiments by putting selective pressures in the bacteria in the lab that that's how that cross resistance exists. Another way that antimicrobial resistance can be transmitted is through phages. So phages can actually, they're really cool, and I don't think I'm smart enough to understand them. I just think they're amazing. And they can pop in and out of bacteria whenever they want. They can be amazing treatment alternatives, but they carry genetic material with them. And sometimes they can carry antimicrobial resistance genes and insert that into bacteria whenever they infect and go lysogenic. And then last but not least, bacteria can actually make direct connections between cells
Hmm.
and transfer genetic material that way. And I'm gonna space out a little bit here because you might appreciate it. And in fact, I have this piece of art that I bought the ASM in 2022 that shows exactly that. So I'm gonna show you that. So what you're seeing in this piece of fused glass art here,
Oh, that's Jane's work.
yes,
That's Jane Hartman's work.
yes. So this is like antibiotic resistance happening, transmission happening in real time.
Oh no, that's a lovely piece of art. I have
Yeah.
some. I don't have quite that nice. But Jane Hartman's a wonderful artist. So this
Yes.
idea that DNA is moving around all the time, and we're used to the idea of transformation, transduction, and conjugation, all of which you've discussed. And then there are also these little membrane vesicles that are more and more important in horizontal gene transfer. And then there are other things where we don't even know the mechanism. The point I'm trying to get at is there's so much horizontal gene transfer, and this is fundamentally different than the way that we look at animals and plants. And so this level of sharing is just remarkable. And it's what is the other part of antimicrobial resistance that we have to worry about. It's really terrible enough when a bacterium becomes resistant to an antibiotic, but the ability to donate that to other bacteria, I can't even say how much worse that makes that potential problem. Worse is my value judgment. To the bacterium, it's perfectly fine.
It's actually a competitive advantage, right?
Hmm.
Sometimes they get that and
Can
they're
be.
better off.
Yes.
So I also find the concept of horizontal gene transfer fascinating, and I don't think people grasp it quite how crazy that is. So I came up with kind of a gross analogy and to try to put the point across. So imagine you went to a funeral of a friend of your family, and you get there and this person had beautiful hair, and you're paying your respects and you decide, oh, you guess what, I love their hair. So you just grab a chunk of their hair as you're paying your respects, and then you put it in your head. The next day you have the exact same hair. That sounds absurd, right? Except bacteria can do exactly that. They can
Hmm.
just pick up pieces of DNA and get those exact same powers, if you will.
Erika, I say it's very similar to like you having children by a Caesar salad. It's that level of difference phylogenetically where you can get gene transfer. And people think that I'm being flippant. Sure, I am. I'm nothing if not flippant, but I'm also accurate here. That level of gene transfer makes this, and remember, this took place long before anyone ever started using antibiotics for any reason. That's the hard part for me to get across to students. This is not in response to antibiotics. This has always been the way the microbial world has been.
Yes, and as you can see on this graph, I really like to point to this study. Antimicrobial resistance is a threat for human and animal health because makes our drugs not effective, and we need to be ahead of the curve in that sense. But bacteria are always winning. They have been doing that for millennia. Antimicrobials and resistance is how bacteria learn to compete and to win environments and evolve to be able to adapt.
Mhm.
And this, the fact that you can find antimicrobial resistance in a certain environment does not mean it's necessarily a bad thing. There
Right.
has been reports of antimicrobial resistant bacteria in ice caps and glaciers, and the key here is we need to understand what the resistance mechanisms are. We need to know which ones are present and how can we use that information to better treat our patients or use that information to better change policy to be able to keep the one health system healthier.
We're part of the planet, and we tend to think of ourselves as being separate from it. So, and then it takes you back to what my late father was saying, that the food didn't like magically appear in the store. So
It's
you'll
like
laugh. You'll laugh. It bothered him so much that he insisted I grow carrots and strawberries in our backyard just so I understood it was possible to do that.
it's crazy how many kids think that milk comes from a carton.
Hmm.
It's like, yeah, it's a big goal of mine is to educate and really give credit to all the hard work the farmers, the 2% of farmers that feed 100% of people in this country nutritious, healthy food.
So I think it's wonderful. You've talked about a couple of different issues here that you're studying in your lab. You're basically doing, and this is important, surveillance to show that there is the evidence of antimicrobial resistance associated genes in particular environments and watching how they change over time. This is important. And it's not that different than how people who are epidemiologists study how viruses move through a population. This is the same kind of thing. And then you also talked a little bit about Salmonella Dublin, which is a really great model system for a number of situations, and not simply a model system, but something of direct benefit to farmers.
And
I have,
humans.
and humans, but because we're all part of the same thing. And this is something that I've said many times. I have never done anything in my career that benefits a human being. Okay. I have only looked at questions that I thought were cool. That's been me. But what you do matters because you're actually helping other people. And I think we forget sometimes that's an important reason why we do science.
That is true. It's just, and not only on the studying bacteria that are causing disease, but I
Mhm.
think one of the best parts of our job as professors is to be able to see people gain new knowledge and learn new things and be independent. So I I absolutely agree. I do, however, veer a little bit more toward the practical approach.
Mhm.
For example,
Yeah.
I'm seeing the farmers struggling with this bacteria. What do we know about it? How widespread is it? Can I make a better diagnostic test for it? Why is it so resistant? What can the farmer do to be able to manage it on their farm better once they get the infection? On the other side, with the antibiotic-free feeding, what are all of these products doing to the microbiomes? Because there is a lot of research on product A or product B, but there is not a lot of head-to-head comparison of different classes of those products at once. And that's one of the questions that we're trying to answer. Going back to the fact that antibiotic resistance is a natural phenomenon, as you can see on this timeline, antibiotic antimicrobials were first discovered in the early 1940s, and it was a revolution. And I like to say that it was a serendipitous discovery by Fleming, who decided to leave and forgot some plates out and came back and found that he left the lab a little bit messy and ended up discovering one of the most powerful drugs that have helped mankind. So between the 40s and the 70s, we have learned and discovered novel antimicrobials, many of them, if not most, produced by microbes themselves because it's a natural phenomenon. And what I want to point out on this timeline is that not much later, or sometimes even before we discover these antimicrobials, we discover resistance in the pathogens that we are trying to treat with those antimicrobials. So it's kind of like an arms race. We're never going to win, but we can
Mhm.
be ahead if we know what is happening and being able to choose the right drugs to treat. But in the end, with everything, prevention is much better than treatment.
Louis Pasteur said, and I'm sure it's better in French, that in the end, gentlemen, he said, the microbes will have the last word. And it's really true. Like I say, first evolved, last extinct. That's the microbial world. And we tend to forget that. So, Erika, knowing what we know and knowing that fewer and fewer antibiotics seem to be working, there are efforts to try and look for new antibiotics, but they're pretty much using the same approaches as before. Do you have any thoughts, knowing what we know about possible alternative pathways to find new antimicrobial compounds?
Yes, Mark. The problem of decreasing and new antibiotics is real, but there are a few paths forward. So one success story that we have is the use of bacteriophages as an alternative to treatment whenever you have an antimicrobial resistant infection. And I'd like to point you to the work of Stephanie Strath-D out at UCSD. I'm not, yes, you're familiar with them, and they have a couple of fantastic videos online. So one of them is use the bacteria enemy against it. So the
Hmm.
enemy of my enemy is my friend. One of the drawbacks is that is like a sniper type approach where you have to be very accurate with which bacteria you have and which phage you have, and that takes a lot of time. And it's really hard to produce in mass. So that's part of the reason why we don't have new drugs. Another thing that have been really transformative in the recent um, years is the use of artificial intelligence. So now we can start mining data sets and try to predict things and generate new hypotheses of, oh, this random gene that nobody knows what it is seems to be very similar to this well-known thing. Maybe we want to clone it into a bacteria and see what it does. And that could end up
Hmm.
with a new antibiotic, a potential new antibiotic. But the biggest path forward is increase education and prevention and really diagnostics. So you need to know what drug you need to use to be effective, and you need to do those treatments appropriately. We can still be very well healthy with the drugs we have currently. It's not like nothing is ever, it's we're going towards nothing being useful ever. It's a matter of you need to be quick to find out which bacteria you're dealing with and what superpowers do they have so you can use something else to treat your infection.
And I really like this concept of the arms race, even though some folks don't like that metaphor.
Mhm.
But remember that when we are using an antibiotic, that's not changing. And so bacteria are mutable, are changeable, are protean. So they're going to get around it however they can. And so I suspect, honestly, and I've said it before, that some of the solution is going to be ecological. You wisely pointed out that that's part of One Health and making sure that everybody is in the whole food supply are healthy. And so, what I believe will happen is we're going to learn that the way that we deal with different diseases, and it could be in the farm or it could be in our gut, is going to be an ecological solution. But that's me.
And that No, I completely agree. And if you think about that ecological solution, that's prevention, right? If you're
Hmm.
keeping a healthy ecosystem within your gut, you're preventing that one pathogen of having the opportunity to outgrow the others and cause disease. So I completely agree. And we can actually use certain types of feed ingredients or lifestyle changes, both in humans and animals, to be able to steer this ecology a little bit in order to keep us healthier. And I'm just super excited to see everything that's coming out in terms of this research and the holobiont concept, where we are not just human beings, but we're a community of organisms that are walking around. And that makes us not that different from cows.
No. I used to make a joke. I say things in class that students pick up on, and they find it funny. Like I'll call students petri dishes with shoes, and I'm not making fun of anyone. We really are. We swim through this sea of microbes at all times, and we tend to think of them, either as angels or devils. And the truth of the matter is, most microbes see us as a surface, if that. So understanding the context of everything is really important. And that's kind of what you have to be able to do when you're doing microbiome work. You're asking the question, how do these change over time or in response to? And it's as complex as the Amazonian rainforest. And it's a beautiful, wonderful thing to think about. And I am not trained as an ecologist, good Lord, no. But it's an interesting way to look at things. And so it's delightful beyond words. I also want to say to listeners and viewers that Dr. Ganda's website goes into so much fabulous detail and is so broad and so interesting and so accessible that I strongly urge people to have a look at it.
Oh, thank you so much. I I think I want to say something that might make more sense in other bits of AMR is one of the things that I do in my class. Have you ever played the gut check game?
Yes, yes.
So we
I
play
think
the gut check game.
yeah, I have a copy of it right here. Yeah.
Yeah, so we play the gut check game in my class, and the students just started realizing that taking antibiotics just in case can actually have like poor consequences because it can disrupt that ecological balance.
Yes.
So another thing with AMR is that we need to tell people that taking an antibiotic just in case is not a good idea from a One Health perspective because it might make your microbes potentially resistant. Just like don't go to the doctor and ask for an antibiotic all the time. Because
And,
that's
and there were physicians for many years that would just to make them quiet, they would give antibiotics. I don't know if you know this story, but I think it's relevant to it. I was treated for acne
Mhm.
as a teenager, and I was on a gram of tetracycline a day for about 6 years. So they had to take blood samples about, every month to make sure I wasn't killing my bone marrow. The reason I bring this up is that many years later, I took the microbial diversity course at Woods Hole with the late, great Abigail Salyers as my instructor. And she was really into bacteria that lived in the gut. And most people aren't interested in anaerobes, but that's where the action is when it comes to the gut. And to make a long story short, we isolated Bacteroides. Yes, they've changed the genus name since, from our own gut. And it turned out that about 30% of mine were still resistant to tetracycline. And that's after decades where I hadn't had tetracycline again. There was a woman in our course who was from Belgium where you can buy antibiotics at that time over the counter. 85% of hers were resistant to tetracycline. So, misuse of antibiotics is a problem. That is not directed toward agriculture. I am saying that we humans do that.
no, everybody can misuse antibiotics. And that's why having the concept of antimicrobial resistance and making people understand what the One Health concept is and how you taking something just in case might actually cause you harm decades later, or
Mhm,
you might be carrying a bacteria that is resistant and you can
mhm.
make your family member sick because you transmitted it to them unknowingly and then they can't get treated. Absolutely.
So there's a really fun demo that I do that you might think of for your students where you spread a plate with E. coli, which is sensitive to ampicillin.
Mhm.
So nothing grows other than maybe occasional colonies. And then you place a tiny droplet or a tiny colony of a bacterium that expresses green fluorescence protein and is resistant to ampicillin because it's secreting beta lactamase.
Mhm.
And what you get after about 4 or 5 days is that central green fluorescent region where you put the resistant bacteria, but all these non fluorescent satellite colonies around it that are simply taking advantage of the fact that the first bacterium was inactivating the antibiotics in the plate. And that can happen in our guts. And it
That is
really,
so cool.
yeah, I think it, and I'll send you pictures. It's a
Yeah.
wonderful, wonderful approach to things.
I really love how you you do the research experiences in your course, and I really
Mhm.
try to bring more of that. And they just care more and get more excited about it.
They do if it's theirs. And that's what the real secret is. We're starting to get a little bit low on time. And so what I wanted you to do is maybe say a couple of things that you're particularly proud of from your research group. We've given a lot of advice to people, and I'm going to put links in the show notes about using antibiotics in a wise fashion. But do you have anything you want to brag is a strong word, but I'm going to use it. Brag about because your research group is awesome?
I like to say that I don't do any of the work. I just beg for money and fight bureaucracy, and they are the real stars running the show.
Hmm.
So I am very happy and feel very fortunate to have been able to assemble such a great team. I don't know what they saw in me, but here we are. We have several graduates that have gone on to positions in government research
Hmm.
and faculty positions and gotten lots of fellowships and awards, and they just do it and they do it well because they care about their research. And the other thing that I like to brag about is our community, pun intended, of people studying microbes at Penn State. I feel like that is a secret sauce of having that critical mass of folks that you can ask advice for and really do that interdisciplinary One Health type approach.
I think that's a really wonderful note to kind of end on because it's wonderful to be able to walk right down the hall and come up with ideas or bounce ideas off people. Not everybody can do that. And I know that what's been done at Penn State is with that in mind. And you're part of that constellation.
Yes, we are an amazing group. We just got an award, which we're all very proud of. And it's not just walk across the hall, we are very engaged across departments and colleges as well. So we work with artists, we work with folks in other campuses, we work with folks in other colleges, and it's just so much fun. Um, we have a fun, completely unrelated story where we worked with folks in information science and technology, and we found a potential new pathogen that can be causing metritis in dairy cows that we haven't really looked for before. Because we did some microbiome work with some fancy statistics, and like, wait a minute, Elcococcus? Really? And then we are further investigating that now.
It's just a matter of looking. And one of the things that I love about the microbial sciences, and I'm not going to say that there aren't from time to time difficult people, there are. I'm not going to say that it's perfect because it's not. But microbiology has always been an extremely welcoming environment that I have seen and others have commented on. And that's one of the reasons that I have pushed is a strong word, strongly urged students to enter that field. And I'm delighted that they are forming a nucleus of a new generation. We often say there's a statement that we need a big tent. That is to say, lots of different people kind of talking to one another, communicating ideas. So in essence, we can talk about a big petri dish because that's what we need is a big petri dish. And you've got one at Penn State.
Yes, we are a beautiful petri dish with lots of smart and humble people.
Well, I want to thank you for this wonderful. I mean, I love the breadth of your interests. I love your enthusiasm. I hope listeners and viewers enjoyed this as much as I have. I am so grateful for your time. I want to wish you and your collaborative team and your friends and family the very best.
Thank you so much, Mark. This has been fantastic. And I have been following your work through Twitter for a long time, and it's really a pleasure and an honor to be interacting with you.
You're very, very kind. Thank you so much. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with awe-inspiring links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Erika Ganda is in the Department of Animal Science at Pennsylvania State University in University Park, Pennsylvania. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.